Embryo grading is what we do in order to select which embryo should be better to transfer in a IVF cycle. Embryo grading has a long history. We used to grade eggs, then we used to grade zygotes. As far as the technology started to evolve, also the time that the embryo was spent in a lab in culture increased. 10-15 years ago, the vast majority of the transfers were done on day 3 of the embryo development, where the embryo should have reached anywhere between 6 and 10 cells. But since then, quite massively worldwide, embryo culture to day 5 or day 6, or until the blastocyst stage has emerged and has been incorporated in current practice. When we're talking about embryo grading at this point, we're talking about the grading of the blastocyst. At this stage, the embryo has already 2 types of cells. One type of the cells that are forming small clumped mass of cells, that's called the inner cell mass, from which eventually the baby will be formed. And the outer cells that are forming something like a globe shape are called the trophectoderm cells. So those are responsible to, first of all, to signal the implantation and then to form the embryonic parts of the placenta. So when we're looking at the blastocyst, this is what we look at. We look at the inner cell mass and the trophectoderm, and also we look at the expansion grade. When the trophectoderm and the ICM start to separate, basically a cavity formed with fluid starts to emerge between them. As long as that fluid starts to accumulate, is gonna make the embryo expand more. Grades 1 and grade 2, for example, they're early stages. So before grade 3, which is called the full blastocyst, was the first stage where you can actually grade the inner cell mass and the trophectoderm. And then the embryo just expands more. It may increases the volume. That's going to be stage 4, which is expanded blastocyst. And when they start to breach the zona that protects the embryo in the first 5 or 6 days, that's called stage 5, when it's hatching.